In this talk, I would like to give you an overview of the research that is being executed at the team of Professor Kathleen Marchal, part of ID Lab. Professor Kathleen Marchal will also join the live Q&A. Let's consider as an example prostate cancer and the challenge to identify successful new drugs and subsequently how to recruit the right test patients in clinical trials. We will discuss two types of experimental setups that are used for the detection of biomarkers and potential drug targets. We will cover the challenges to apply traditional data analytics and introduce you to our R&D on graph-based data mining. And for the two types of test setups, we explain the benefits of our approach. The first experimental setup to drive R&D in pharma is based on cohort analysis, meaning the study of a group of patients that are being treated in standard of care for similar disease characteristics and for which samples of individual tumors have been taken and sequenced. Then the sequence data is typically molecularly profiled and the DNA or genetic data is associated with the observable characteristics of a disease. The latter is called the phenotype and captures the interaction with the environment. To make it a bit more clear, for an identical twin, the genes are identical, but it is the phenotype that will declare the differences in reaction to a disease. What you see in this picture is that in such a study you typically have two patient groups. The metastatic patients where the disease is well managed and the non-metastatic patient group where the disease is still out of control. From such a study, causal molecular entities are being searched for, either to identify potential new drug targets or to identify biomarkers that then can be used to stratify or cluster patients for clinical trials. These studies here are just based on real-world data and help to understand the mechanisms and drivers that underlie the disease. A second type of experiments to perform R&D on next-generation drugs is called gene knockdown cell line screening. Here we do not use direct patient material, but cell lines that are derived from patient material and that model a certain disease. Cells are cultivated, here as an example the prostate cancer cells, and during gene knockdown, the effect is studied with the disease characteristics. If the knockdown alleviated the disease characteristics, then the identified gene is a potential drug target. There are very obvious target candidates, so the more dependent ones on the slide, the ones in the red color here. And however, nowadays we are also more and more interested in the less obvious candidates. This cell line screening method can be used to model and generate data. It is easy to cultivate and easy to read out and assess the severity of the phenotype. So it's a second method to facilitate building of drug discovery pipelines. Both experimental setups facilitate drug target prediction. However, the challenges we are facing is that data are expensive to generate. So there is a need to come up with cost-effective ways to navigate towards the next drug candidate. We also need to account for the fact that data are still small in statistical terms, and we are dealing with underdetermined problems. Data is often noisy and also incomplete. So that's why the team of Professor Kathleen Marchal has been working on integrative approaches to cope with noisy and incompleteness and underdetermination, and to identify targets with lower signals that were not visible before. Why are we using graph-based approaches? It is because graphs can intuitively capture known information on molecular interactions between genes, which can be used as prior information to steer the mining of the data in a way that reduces the underdetermination of the problem. And also, graphs allow you to capture relations that are biologically relevant between different data sets, and that then can help you in defining mining strategies that are more interpretable. To illustrate the level of underdetermination, we use an example of cohort analysis. What you see here in the dark bars are molecular entities that became aberrant by multiple mutations. Pharma is typically interested in identifying those aberrations that occur more frequently in the group of metastatic patients, because these are the ones that might be causal to metastatic disease and they can interfere as potential drug targets. Here we have to realize that the number of causal mutations is largely outnumbered by the total number of mutations. It is a ratio 3 to 1000. So you are searching for a needle in a haystack, and the number of independent samples you have is typically limited by the budget, as molecular profiling is very expensive. Cancer is a pathway disease and therefore a complex disease. You can have different patients with the same phenotype behavior, but where aberrations occur in the same pathway, but through a different genetic entity. The result is that you end up with statistical signals that become very sparse, so you can no longer use classical statistical approaches. To deal with this complexity, we are applying graph-based approaches that incorporate prior information in the form of interactions between genetic entities, and we try to identify not single entities that are recurrently mutated, but combinations of entities that are closely connected in this interaction graph. This way we can increase the power of identifying a true signal. Here you see a public data set on prostate cancer. It's the Hartwig data set. What we did here is we combined this with prior information on genetic interactions and applied the graph-based probabilistic pathfinding approach that is able to work on very large data sets with many patients and mutations. It allows to extract the pathways that are causal to the origin of metastatic prostate cancer. And the key message here is that we were able to demonstrate discovery of rare mutations. In a biological relevant way, we managed to extract driver subnetworks from large data sets. This is thanks to the fact that we managed to include prior knowledge and can attach probabilities to every node in the graph, outperforming, for example, random walk strategies. The second example relates to the cell line screening experiments and illustrates how data can be noisy. Each row here is a different cell line knockdown experiment, and every bar is an experiment of thousands of euros. So by transforming the data with the graph-based link prediction model, we demonstrated that we can exploit prior information to make more reliable decisions in the intermediary range of less dependent genes on whether the gene has an impact or not. As a conclusion here, a major benefit is that we can start predicting for genes for which no knockdown experiments have been performed yet, and in particular we can predict what the impact on the disease will be. This methodology can thus be used to design experiments and to drastically reduce overall costs in drug discovery, and through relevant biomarkers improve patient recruitment in clinical trials.